All right, so this is the second lecture for Chapter 16 in Unit 3, Metabolism. And in this lecture, we're going to talk about glycogen storage diseases. So this is sort of a clinical application of glycogen metabolism, which we talked about in the previous lecture. These diseases are all very high yield for biochemistry exams. Also, definitely can show up on board exams as well. So this is really important content. So first, just a general overview of glycogen storage diseases. They all result from some kind of defect in carbohydrate metabolism. So it's a defect in one of the steps that we've talked about in previous lectures. What they result in is glycogen accumulating within the liver, the muscle, or both. Because remember, those are two major storage points of glycogen, liver and the muscle. And then they can be identified using a special PAS stain on histology, because this stains for carbohydrates. So if you have a buildup of glycogen, which is a carbohydrate, you're going to be able to stain for this. This is something that could show up on a question stem that could help you point towards a glycogen storage disease. So it's something to be aware of. Last thing here is that liver defects, because remember, glycogen storage and glycogen synthesis and breakdown is important processes in the liver, especially since the liver provides free glucose for a lot of peripheral tissues. If the liver defects will result in decreased gluconeogenesis, because remember, this happens mainly in the liver, can also happen a little bit in the kidney, but mainly in the liver. So if you can't carry out gluconeogenesis, you can't produce free glucose. And so what that's going to lead to is hypoglycemia. And so you'll develop symptoms of hypoglycemia mainly during fasting, because if you consume glucose, you certainly have a store of it there. But during periods of fasting, you're relying on gluconeogenesis to provide that glucose to carry out glycolysis. So the first one here, type 1 von Gurkey's disease. So this involves the deficiency of glucose 6-phosphatase, which is actually an enzyme involved in gluconeogenesis. So it's defective in the liver and the kidney. And what's a small little fact we threw in here is that glucose 6-phosphatase is located on the endoplasmic reticulum. That's not quite as high yield, but it's a nice little fact to know. The inheritance is autosomal recessive. And so what will come down here, this is our diagram for glycolysis. And if you remember, when glucose gets taken up into the cell here in the liver through the GLUT2 transporter, first thing that happens to it is you phosphorylate it so that you can contain it in the cell. So you convert it to glucose 6-phosphate. However, during fasting, especially in the liver, remember, you're trying to provide free glucose for peripheral tissues. So you'll break down glycogen, you'll convert to glucose 6-phosphate. Also, through gluconeogenesis, you're going to convert that eventually to glucose 6-phosphate. Then the last step for both of these to give yourself free glucose is you need to take this phosphate off the 6 position here and convert it back to glucose. The enzyme that does that is glucose 6-phosphatase. Now, if you don't have this enzyme, you're going to be unable to convert glucose 6-phosphate to free glucose. So in theory, these processes can still occur, gluconeogenesis, glycogen breakdown. The problem is that in the end, they don't carry out their end goal of giving you free glucose. That's the key. So what it really leads to is a severe buildup of glucose 6-phosphate, and we'll talk about why that's important in a second. And so remember, you want free glucose so you can export it to other tissues. So as a result of not having free glucose to export, you're going to develop hypoglycemia. So you'll have low glucose levels in the bloodstream. A byproduct of this is it leads to a buildup of lactic acid, which can give you lactic acidosis. And it can also give you high levels of uric acid, which can actually then result in gout in these patients. We'll explain why these happen on the next slide. So on this slide, we're going to talk about the clinical features of von Gurkey's disease, but we're also going to tie in the metabolic reasons for why these show up, because that can really help you understand this disease versus just memorizing the different components. By understanding the biochemistry and the metabolism and the reasoning behind why these clinical signs and symptoms show up, it can make this disease much easier to understand and much easier to remember on test day. So the main one here is hypoglycemia. So remember, we said on the last slide, what you're going to end up having is a buildup of glucose 6-phosphate, because what you would want to do with that is convert it to free glucose, right? That can then be exported to other tissues. Now, if you're not able to do that because glucose 6-phosphatase is absent, so if you can't convert it to this, you're going to have a decrease in free glucose, which is going to lead to hypoglycemia. And a hypoglycemia is going to lead to seizures clinically, and that's a important manifestation of von Gurkey's disease. You could definitely see that clinically, and you could see that pointed out in a question stem. Because again, the way they're going to test you on these is they're going to give you the clinical symptoms in the stem they're not going to say it's von Gerke disease. They're going to give you the symptoms in the stem, and you're going to have to be able to deduce that it is von Gerke's disease and then answer some kind of either secondary or tertiary level question as a result of that. And then a real serious complication of this whole cycle here of hypoglycemia, seizures, is you could actually end up with hypoxic brain injury as well. Now, if you have a buildup here of glucose 6-phosphate, one route it can go is it can then get converted into pyruvate, as we show here, through glycolysis. So if it's going to build up, it's going to go somewhere. And so if it goes to glycolysis, then you'll have a buildup of pyruvate. And then if you have a buildup of pyruvate, that can get converted into lactate, which gives you a high level of lactate, which can result in increased serum lactic acid or a lactic acidosis. The other driver behind this is that glycolysis, remember, produces a high level of NADH. And so if you're producing high levels of NADH, you're going to have depleted NAD+, because remember, you need NAD+ to then produce NADH. So in the cell, it's to the cell's advantage to regenerate this NAD+. So that's going to drive the conversion of pyruvate to lactate as well, because it's going to give you NAD+ that can then be used in glycolysis and other processes within the cell that rely on NAD+. Another result of increasing pyruvate is that some of the pyruvate then gets converted into acetyl-CoA. Acetyl-CoA can then go be used to produce fatty acids and cholesterol. So on the serum labs, you can see increased serum triglycerides, and then what that can result in is actually xanthomas and even a fatty liver as well, clinically. And so that's the reasoning behind it as well, is it all stems back to this buildup of glucose 6-phosphate, which leads to a buildup of pyruvate, which can either go towards a buildup of lactate or a buildup of acetyl-CoA, leading to buildup of fatty acids and cholesterol. The other thing is that glucose 6-phosphate can then go back to producing glycogen. So if you have a buildup of this, it can go towards producing glycogen. So if you have a buildup of glycogen, that can lead to hepatomegaly because it's taking up space in the liver and that causes it to enlarge. Another sign you can see is enlarged kidneys as well. And then here you may be wondering how they can develop gout. And so let's talk about that. So you remember gout comes from increased uric acid. So if you have increased levels of uric acid, that's going to give you gout, right? So the other route, and we haven't talked about this yet. This is going to be in a subsequent lecture from this, where we'll talk about the pentose phosphate pathway. And this is the pentose phosphate pathway here, and we show this. And for the purpose of von Gerke disease, we're going to really show you just kind of the main couple things you need to know from this. You don't need to know this whole process yet to understand this disease. So you have a buildup of glucose 6-phosphate. Another direction, so it can go towards glycogen, it can go towards glycolysis. Another direction it can be shifted towards is the pentose phosphate pathway. So you can see that here. So it'll enter the pentose phosphate pathway, and one of the results of pentose phosphate pathway is producing ribose 5-phosphate, which is then used in nucleic acid synthesis, specifically purine synthesis. So if you have a buildup of purines, so you increase purine synthesis, that's going to lead to a buildup of uric acid, because remember we talked about purine synthesis in Unit 1. So increase purine synthesis, increase uric acid, and that's can give you gout eventually. The other things here, just real quick, you can have platelet dysfunction that results from this. You can have an enlarged abdomen, and these patients can also have short stature and doll-like faces, and these are just also results of the metabolic dysfunction that's occurring here. And so, again, these are all kind of more buzzwords here, but remember, these are a result of this impacts an individual from day one, and so it's unfortunately affecting them as very young children, and so you'll see this manifest in very young children often. The way you treat this is, remember, they can't really produce free glucose from the liver during fasting, and so what you need to do is constantly be supplementing directly with glucose to help make up for that, to help control their symptoms and, help feed their metabolism. All right, so type 2 Pompe disease. So this involves lysosomal enzymes involved in glycogen metabolism. So it's a deficiency mainly in alpha-1,4 glucosidase, which is an enzyme found in the lysosome, and it's analogous to debranching enzyme, which we talked about in the previous lecture, which is responsible for breaking down these branches off of the main strands of glycogen. So it has a similar function to that, but it's for glycogen stored in lysosomes specifically. Now, it also has alpha-1,6 glucosidase function. This enzyme here also has this function. I would make this correction in your book. It's not like it has both these enzymes and you're deficient in it. If you're deficient in this enzyme, that's a different disease we'll talk about in a few slides here. So really, it's a deficiency of this enzyme, which is a lysosomal enzyme. This enzyme in the lysosome also has alpha-1,6 glucosidase function. So I just wanted to make that clarification. Apologize if there was any confusion with that. So this is the main enzyme that's deficient, and we indicate that down here. So alpha-1,4 glucosidase, Pompe disease here, and its inheritance is autosomal recessive. And so what happens here is, since you're not able to readily break down glycogen within lysosomes, what happens is you have an increase in lysosomal glycogen storage. And so these vacuoles of glycogen will accumulate within organs, and what that does is it can affect cellular function, and then it also holds up some glycogen from being used readily. And where that really comes into play is high energy organ systems, or high energy organs that really rely on readily available glycogen very quickly and consistently throughout its physiological function. A very good example of that is the heart. It's pumping all the time, 24/7. Always needs energy readily available in the form of glycogen. And so that's where it affects the heart very, very significantly. And then also affects skeletal muscle, because skeletal muscle, at the drop of a hat, you could be running, going from a resting state to running, or, actively moving. And so you need to be able to break down glycogen and get glucose readily available. So it affects that. And then the liver also is just a major hub of glycogen storage and glycogen breakdown, and then also being able to provide glycogen for other organs. Now, since you still have other stores of glycogen available, what can happen here is it can still result in relatively normal levels of glucose. What I would add to this is you can also have mild hypoglycemia. And this is where you're able to differentiate it versus von Gerke disease, which we talked about in the previous slides. And so von Gerke disease, you have very severe hypoglycemia because you can't even carry out that last step of gluconeogenesis or glycogen breakdown to free glucose, and so you have very decreased levels of free glucose. In Pompe disease, you either have relatively normal or mild hypoglycemia, so you don't have those severe hypoglycemic symptoms that you may see in von Gerke, such as seizures or other symptoms like that. All right, and so the clinical features are directly related to the impact this disease has on those high metabolic organs, such as the nervous system, the heart muscle, skeletal muscle, and so on. And so retardation, that's obviously relevant to the nervous system. This is a disease that it's an inherited disease, so it affects the child from day one. So it affects with neurological development. Also, by affecting metabolism, and the ability for neurodevelopment requires a great deal of energy, and so by affecting metabolism, that can impact that. Hypertonia, again, that's due to skeletal muscle. So impacting skeletal muscle. Hypertrophic cardiomyopathy, that's obviously due to cardiac muscle. And then cardiomegaly, eventually it can lead to that. Now, another thing that symptoms you can see as a result of skeletal muscle and then having cardiac muscle affected, you're not able to get adequate perfusion, and then also skeletal muscle, you're weak, you develop weakness. So you can have dyspnea, so shortness of breath. You can develop those as well. Exercise intolerance. And again, this is related to both cardiac function and then also skeletal muscle function as well. So the symptom you may see in a STEM is that a patient gets easily fatigued while trying to exercise, because again, remember, exercise is going to be increased metabolic demand. And again, that's what states or tissues where this disease really impacts the patient is when they're trying to carry out certain functions that require high levels of metabolism. So exercise would definitely be one of those. So exercise intolerance, and then what these can eventually lead to is heart failure. And unfortunately, this develops much, much earlier than in the typical adult population. And so unfortunately, what this leads to is early death often. So moving on to type 3 disease, or Corey disease. This is a deficiency of alpha-1,6 glucosidase, which is also known as debranching enzyme. We'll do a quick review of that, of debranching enzyme, in a second here. This is an enzyme that's especially present in muscle and liver, and it's an autosomal recessive inheritance. So if you remember this, we talked about this in the previous lecture in glycogen metabolism. So you have these primary strands here of glycogen, and then you have these branch points. So remember, these are those 1,4 alpha 1,4 linkages, and those are those alpha 1,6 linkages that allow for branching. And remember, branching allows you to more efficiently store glycogen, also allows you to more efficiently break it down, which, as you can see, is very relevant in this disease. So when you're breaking down these branches, you start out at the outermost here, and you're breaking down these 1,4 linkages via glycogen phosphorylase. So glycogen phosphorylase can break them down, but then when it reaches this point where you're 4 units out from the branch point, glycogen phosphorylase cannot carry out any further breakdown. That's where debranching enzyme comes in. So debranching enzyme takes these 3 units here and then essentially adds them, as you can see here, to the primary chain here, leaving this one single unit here. Now, in Cory's disease, this is defective, so you can't take this and you're not able to form this. And so what you're left with is abnormal glycogen structure with many of these short chains like this. So you have many of these like this, and these are units you're not able to break down further into free glucose. So you're limited on the amount of glycogen you can break down. Makes it much less efficient, much less effective. And so that's what leads to the clinical manifestations of Cory disease. And the main one here would be hypoglycemia. Not as severe as von Gerke's disease, but still pretty significant. And so you definitely will see hypoglycemia. The other thing this leads to, in addition to not being able to break down glucose efficiently, is it also leads to significant glycogen buildup in many major organs. And that would include organs such as the liver, muscle, skeletal muscle, and even in some cases the heart. Now in the liver, if you have buildup of glycogen deposits in the liver, that will lead to hepatomegaly, so enlarged liver. Now the other thing here we list here is increased uric acid. That's not, I would actually cross that out. I apologize about that. That's not entirely true for Cory's disease. That's more seen in von Gerke's disease because you don't have that buildup of glucose 6-phosphate like you do in von Gerke's disease. So you're not being pushed over to that pentose phosphate pathway. So you're still having, obviously gluconeogenesis is okay. You can still provide glucose via gluconeogenesis in these patients. They do have elevated levels of triglycerides, however, again, not to the degree of von Gerke's disease. And then lastly, they also have normal levels of lactate as well, because again, they're not having that excessive level of glycolysis, which is leading to those high levels of lactate that you see in von Gerke's disease. So they do have gluconeogenesis, normal levels of glycolysis. The big thing here is they're just not able to efficiently break down these branches of glycogen. And so it leads to buildup of these glycogen deposits in many of the organs, and then also leads to mainly hypoglycemia because you're not able to break down as much glycogen as you'd like to. Later on, as this disease progresses, what it can lead to is more skeletal muscular disease, where you would see hypotonia, and then eventually you can lead to deposition of the heart and dysfunction of the heart, which can then lead to cardiomyopathy. So again, the major ways you would just differentiate this from von Gerke disease is the normal levels of lactate, not as severe hypoglycemia, and not as severe levels of triglycerides as well, elevated triglycerides. And then versus Pompe's disease, remember that really affects skeletal muscle and then obviously the heart immediately. This doesn't really affect the heart until later. And so exercise intolerance is a major one where that really would clue you into Pompe's disease versus not so much as for Cory's disease. So type 4 Andersen's disease is where you have defective glucosyl 4,6 transferase, which is more commonly known as branching enzyme, which is present in the liver and in the spleen, and I would say in muscle tissue as well. And we'll briefly review here what branching enzyme does. So if you remember, you have the primary long strand here, and branching enzyme can act on, let's say you're forming a really, really long branch here. It can act on here, as we'll show in a second, or it can also take from another very long primary strand as well, and then add it to another primary strand to form a branch. So what we show here is it takes part of a long branch here. Again, it could take from another really long strand, and then adds it as a branch and starts a new branch by forming a 1,6 linkage here. Remember, these are all 1,4 linkages, alpha 1,4 here, 1,4 here, and then throughout here, and then these branch points are formed by alpha 1,6. Now, if you're unable to do that, what ends up happening is you have very abnormal glycogen structure, and you have many long chains, as we show here, with very few branches. And so what this leads to is very insoluble glycogen structure, and it causes severe glycogen accumulation. And so this is especially significant in the liver, the heart, and then in skeletal muscle as well. And this is what leads to the clinical signs of this disease. And so with skeletal muscle, what you'll have is, weakness, because by building up this glycogen, you severely limit the function of the organ that's affected. So for skeletal muscle, you have weakness. And then for the liver, you'll obviously have liver enlargement due to accumulation of these glycogen stores. So that'll lead to hepatosplenomegaly. And then eventually that's going to disrupt liver function to the point of leading to cirrhosis. And then eventually that's going to progress to liver failure and, unfortunately, progress to early death. Patients can also develop cardiomyopathy, but what's more commonly the unfortunate fate here is cirrhosis and then liver failure and early death. The other way you can distinguish this from the others is, again, Pompe disease. The big thing there is exercise intolerance. Von Gerke's disease is very severe, so we'll write this up here. So type 1, severe hypoglycemia. Type 2 is Pompe's, so exercise intolerance, and then also heart failure. And then type 3 is almost like a more mild form of von Gerke's, so you have mild hypoglycemia. And then here with type 4, you don't have those symptoms of severe hypoglycemia as these others. And then really what it leads to is, you have this muscle weakness, but what's very notable is they'll have cirrhosis. And then the way, this could show up on a test is, they could give you a liver panel in the lab section of a question stem, and that can indicate a severe liver damage or liver failure. And so that may tip you more towards thinking Andersen's disease, especially if they have a more normal level of glucose. Type 5 disease, myocardial disease. So this is where you have muscle glycogen phosphorylase is defective. And in the muscle, it's actually known as myophosphorylase. So just be aware that on a test, you could see where the enzyme is actually listed as myophosphorylase and not muscle glycogen phosphorylase. Another key distinction here is that you have normal levels of hepatic glycogen phosphorylase. And so that actually means you have normal serum glucose levels. Because remember, hepatic glycogen phosphorylase is responsible for breaking down hepatic glycogen, which is then used for peripheral tissues. You'll see why that's important in a few slides here. So the inheritance, autosomal recessive. So I thought briefly we'd review muscle glycogen phosphorylase. So here you have skeletal muscle tissue, and remember glycogen phosphorylase breaks these alpha 1,4 bonds. So here it's starting in a branch point. It could also break these 1,4 bonds here. So it breaks them into glucose 1-phosphate and gets to this point. And remember then these guys are converted into glucose 6-phosphate, and in the muscle, they're not leaving the cell. They're then just immediately going into glycolysis so that you can produce ATP, power the muscle and power muscle contraction, especially for exercise. So in this disease, what happens is glycogen cannot be broken down, and it results in a buildup of glycogen in the muscle. Like we said, you had normal glucose levels. This is really important. because, again, they have normal hepatic glycogen phosphorylase. So if you remember when we talked about glycolysis during exercise, so it ends up using up a lot of oxygen, very high metabolic demand. And so by using up a lot of oxygen in normal people, you switch to anaerobic respiration because oxygen's the limiting factor, and you end up building up a lot of lactate. And remember, that leads to muscle cramps and things like that. However, in these patients, they actually have normal levels of blood lactate during exercise, and we'll explain why that is in a second. And then even after exercise, they have low lactate. So we'll explain why that is in a minute. So you have glycogen, breaks down to glucose, right? Glucose goes through glycolysis. This is all in the muscle, by the way. Glycolysis then gives you pyruvate. And then pyruvate can go to lactate, which is anaerobic, or it can go to form acetyl-CoA, which then enters citric acid cycle, which then enters electron transport chain, which uses oxygen and produces ATP. Okay? So in normal individuals, this gets used up. This is the limiting factor because you use up so much of this during exercise. And so what ends up happening is you shunt glycolysis towards producing lactate because you just don't have enough oxygen in a normal individual who's doing extensive exercise, using up a lot of oxygen. You just don't have enough oxygen to run the electron transport chain to produce ATP, so you got to run towards lactate. However, in individuals with McArdle's disease, in the muscle tissue, they're not able to efficiently break down glycogen. And so the level of glycogen is actually appropriate for the level of oxygen. Even though you still have decreased levels of oxygen, that doesn't change. You have significantly decreased levels of glucose available to go through glycolysis. So what that ends up doing is, since you have such a decreased level of glucose, which is then not affected by this decreased level of oxygen during exercise, it actually causes it to shunt towards aerobic respiration and use up whatever oxygen is still remaining. And so that's what leads to low levels of lactate after exercise. Now, what you do see is you still see fatigue and muscle pain during exercise because even though you don't build up high levels of lactate, you're still not producing that much ATP. And so as a result of having decreased glycogen capacity to break glycogen down to give you more ATP, that causes fatigue, muscle fatigue, because there's just not enough energy, leads to muscle pain, and then obviously cramps. And the other thing is you have myoglobin in the muscle, which is helping to carry oxygen. In these patients, this is characteristic. You'll see myoglobinuria as well. And again, these symptoms get more severe with exercise. Now, what's something interesting is these patients uniquely show what's called a second wind phenomenon. And that's where if they kind of push through this fatigue and pain, they almost experience a recovery after 10 to 15 minutes. And the reason for that is that, remember in our general scheme diagram of showing metabolism, essentially at a 30,000 foot view where we have all the processes working together, is remember you can kind of switch to other sources of metabolic fuel. So instead of using glucose, glycogen, the muscle then switches towards using fatty acids or even protein to then feed acetyl-CoA, and then you get acetyl-CoA from these other sources, which then goes through the citric acid cycle and eventually produces high levels of ATP. And then that gives you kind of this second boost or second win to keep going and then have more tolerance for exercise after pushing through about 10 to 15 minutes. So that's definitely, very characteristic of McArdle's disease. All right, and to finish this out, we have type 6 here, Her's disease. And so this is where you have liver glycogen phosphorylase is defective, so muscle is normal. Inheritance is autosomal recessive. And the clinical features here, I'll jump down here and say it's very similar to von Gerke's disease, but much less severe symptoms. So much similar symptoms, but to a much less severe degree. So if we look at our diagram here, this actually helps, even though this is kind of metabolism at a global view, it helps with explaining this disease here. So obviously you have glycogen here that can be broken down into glucose 6-phosphate. However, in the liver, you don't have this luxury here where you're breaking down glycogen. And so what happens is you're not able to convert this back to free glucose, so you have significantly decreased glucose. So you have hypoglycemia, but again, not quite as severe as von Gerke's disease, because remember, von Gerke's disease affects gluconeogenesis, versus in these patients, you have normal gluconeogenesis. That's a big difference, because von Gerke's, remember, it's kind of a double whammy. You have decreased glycogen breakdown, and decreased gluconeogenesis. So back to this. So what ends up happening is you have this buildup of glycogen. So you have buildup of glycogen here, and that's going to lead to hepatomegaly, because it's just, if you have a buildup of glycogen, it's taking up space, it's going to enlarge the liver. And so since you have this prolonged period of hypoglycemia, as far as the body's concerned, it's in a starvation state or a severe fasting state. So it's got to look towards other sources of metabolic fuel. And so as you can see, you could see a breakdown of triglycerides into fatty acids. So you could see increased levels of fatty acid, increased levels of fatty acid oxidation to give you that acetyl-CoA that can then go on and enter the citric acid cycle and then power the electron transport chain to give you ATP. However, if this goes on long enough and it's severe enough, the body may actually then shunt the acetyl-CoA towards producing ketone bodies, which is then you could have ketosis as well, in addition to hyperlipidemia. So you have these similar kind of lab values to von Gerke's disease, but since you still have gluconeogenesis, the hypoglycemia is not as severe, and so it's not driving as severe of hyperlipidemia or ketosis. So this is just a final summary to just kind of point out the unique characteristics to help you differentiate all of these diseases from each other. So again, von Gerke's disease, remember the big thing here is you have that severe buildup of glucose 6 phosphate, because you can't do gluconeogenesis, can't do glycogen breakdown. So this leads to obviously severe hypoglycemia because you can't produce that free glucose. And then remember, glucose 6 phosphate, you're going to have increased levels of glycolysis. So that leads to high levels of lactate. Glycolysis is going to end up giving you pyruvate, which will then go towards acetyl-CoA. Acetyl-CoA can then go towards producing triglycerides or ketone bodies or ketosis. And remember also, glucose 6 phosphate can go towards the pentose phosphate pathway, which produces purines, which results in high uric acid levels. And so that's what gives you that. Pompe disease, remember, this is the one where it affects the organs of high metabolic demand, such as the heart and skeletal muscle. So, and then you still have mild hypoglycemia because you're just not as able to efficiently break down glycogen. But where you really see is exercise intolerance, and then these patients develop cardiomyopathy. Type 3 with Pompe disease, again, more mild hypoglycemia, normal lactate levels, normal uric acid. And in this disease, you'll also see increased levels of triglycerides as well. Remember, type 4, this is where you have Andersen's disease, and this is where it really impacts liver structure and function, eventually leads to cirrhosis, which develops into liver failure, unfortunately. Type 5, McArdle's. Remember, this is where muscle glycogen phosphorylase is defective, so you still have normal serum glucose coming out of hepatic glycogen levels, normal lactate levels as a result of that. However, where this is really felt is in muscle pain during exercise, also can see myoglobinuriria during exercise. And these patients then, remember, they can get that second wind phenomenon because they switch to those other metabolic fuels such as fatty acids or protein when they kind of push through this pain and they have prolonged exercise. And then type 6, Her's disease, again, kind of a milder version of type 1, still have hypoglycemia, maybe ketosis could show up, not always. And then also increased levels of triglycerides, but again, not to the degree of von Gerke's disease. So again, very similar to type 1, but less severe. So these are just kind of some characteristics, things we pointed out when we went through each one of these, but just to kind of see them all together here so that you can help distinguish these diseases. All right, so that wraps up our discussion of glycogen storage diseases and glycogen metabolism in general. And so now we're going to move on to gluconeogenesis in the next lecture.